You're listening to BioTalk with Rich Bendis, the only podcast focused on the biohealth capital region. Each episode, we'll talk to leaders in the industry to break down the biggest topics happening today in biohealth.
Hi, this is Rich Bendis, your host for BioTalk. And as you know, we interview very interesting people within the biohealth capital region from different disciplines. And I'm going to be honest, this is the first time I think that we've had a pure scientist from NIH on our show, who is the Chief of Molecular Development of the Immune System Section of NIAID, which is the National Institute for Allergy and Infectious Disease. That's a long title. But I'd like to welcome Dr. Michael Lenardo to BioTalk.
Thanks, Rich. I'm delighted to be here.
Thank you for coming. And I'm really excited. We had some pre-discussion that's probably going to be more interesting than what we're going to talk about in this interview that we're going to follow up on, but we might hint to some of those things during our discussion today, because I think we are of a like mind of things that are very interesting to both of us with all of these fantastic resources we have in what we call the biohealth capital region, which is Maryland, D.C., and Virginia, and NIH is right in the center of this. So let's talk a little bit about your background. And Dr. Lenardo, you've been around doing research for a while, but you didn't start out doing that in your career early. So let's go back to education-wise. Where'd you get your interest? How do you evolve to where you are today?
Well, it's interesting. My interest in science and research and application to medicine really started in Maryland. I went to Johns Hopkins as an undergraduate, was able to do work at the medical center there and stimulate my interest in genetics, human genetics. And there were some very charismatic and amazing researchers at Hopkins that inspired me. I then went to medical school at Washington University in St. Louis, which is also a very research-oriented medical school. They've had a large number of Nobel laureates that have come out of that institution. So my interests were piqued further. And then after a little bit of clinical training, I decided really to completely pursue research and essentially give up clinical practice of medicine. Went to MIT, worked with David Baltimore and Philip Sharp. They're learning molecular biology of mammalian systems, gene regulation. And when I looked around for independent investigator positions, I found a very attractive opportunity in the intramural program of NIH and NIAID. And I came here in 1989, so this past summer, it was 30 years ago. And I have run a laboratory in the intramural program at NIH that entire time, advanced up the ranks to the level of sort of a full professor type investigator. It's been incredibly rewarding, and really, I think the stuff we've been able to do is a testament to the institution of NIH and what they offer people to be able to accomplish scientifically.
Before I dig a little deeper into NIH, I love your WashU background. I grew up in Kansas City a lot. So I have Midwestern roots, too. And you've got some Midwestern roots, too. So that's why we're both grounded. And we
I didn't mention I grew up in Chicago before coming out to Hopkins.
Super.
I am a Midwesterner.
So there's a lot of people listening that probably don't know a lot about NIH. And then you used a term intramural. And so you're part of an intramural program, but there's an extramural program. And also you're with one institute within NIH. And maybe just for the listeners, if you wouldn't mind, give a little background on sort of the structure of NIH, intramural, extramural, and how many different institutes there are and how they break down the different institutes.
So the history of NIH started with a single laboratory on Staten Island, and over the years it grew into an institute mainly for infectious diseases, tied to some degree to the military and through the public health service. And it was in the 1930s that it moved from a single building being the National Institute, singular, downtown, and a building that was right on what's now the steps of the Kennedy Center, up to the campus in Bethesda, which was donated by a wealthy individual, Mr. Wilson. And when it took root there, it began to differentiate beyond one single institute into a number of different institutes that cover different disease entities. And with an increase in funding, they began to realize that they could support more research than could physically take place on the campus. And that gave rise to the division between the intramural program, which are actually the laboratories within NIH, federal laboratories, and the extramural grant program, which supports now research all over the country. And as the budget has grown, roughly, I would say 89 to 90% of the total goes into those extramural grants and contracts. So all of the medical schools, research institutes across the country, even some overseas, are supported by that portion of NIH. But there's still a large commitment to our own intramural laboratories, which are funded directly from the Treasury.
The size of the institutes varies dramatically from relatively small to very large. And I think NIAID is the second largest institute, if I'm not mistaken.
It's about $5.8 billion.
$5.8 billion, National Cancer Institute being first, and then maybe the third might be National Heart, Lung, and Blood Institute.
I think that's correct.
So you're in one of the more prominent, larger funded institutes, which covers a broad spectrum. If you look at allergy and infectious disease, that seems narrow, but if you really look at the research that's going on there with $5.8 billion, talk a little bit about the breadth of that research.
Well, the research covers everything involving the immune system. So how we produce inflammation in the body, how we fight infectious diseases, autoimmune diseases, allergies, and other atopic conditions such as asthma. All of that involves components of the immune system either not working well enough or working too well and causing overreactions. And so it's very interesting. A number of things, I think, have catalyzed the growth of the Institute. One is that there has been a striking increase in autoimmune diseases and allergies since the Second World War. And we don't understand precisely why that is. But when I was a kid with an allergy was a pretty rare thing. I think you'd be hard-pressed to walk into an elementary school classroom today and not have a quarter of the kids with various kinds of allergies. And also, the other big force, I think, are the fact that we're now so mobile across the globe that infectious diseases that might have been restricted to one particular continent or even one particular country now are spread all over the place. And I think SARS is a good example of something that began in China, was brought over to Canada, reached parts of the U.S., and populations that are not exposed to pathogens are perhaps the most susceptible. So with this great increase in global exchange of people, we see more outbreaks, more infectious diseases spreading. And so therefore our response as a federal government to protect the health of our citizens has to be stronger and more capable.
So I guess this is a very strong vaccine corridor in our backyard here.
Yes, of course.
And also BARDA is focused here. And is that focused within your institute?
Yes. So there's a very strong, and there has been really for decades, a strong commitment to producing new kinds of vaccines. During Bill Clinton's presidency, he started at the NIH in our institute something called the Vaccine Research Center, which gave it even more prominence and more funding to produce new vaccines. It can be done very quickly now with different molecular tools. And I think that is a very important focus of what we do because we can deploy people to get on top of infectious diseases perhaps faster than any other agency in the world.
We've talked about disciplines outside of your primary area of responsibility. Let's talk about yours now, which is your section is the molecular development of the Immune System Section, which you lead, correct?
That's correct.
Tell me a little bit about that section.
Well, I think the name is sort of a funny name. It started because when I got to NIH, I was very intrigued by questions about how the immune system forms different kinds of cells that have specific jobs, and how's that controlled at the molecular level by different gene regulatory events. And as my career has gone on, two things have changed. One is that given the fact that in my building we have a connection to the research hospital on the NIH campus, I've gotten very interested in problems that involve direct translation of basic science discoveries within my lab right to the bedside. And so that has moved my research more in the direction of tackling disease problems such as autoimmune diseases. The second thing that's happened is, given my molecular background, I've been really excited by the new genomic technologies that exist to probe genetic problems that cause immunological diseases. And again, being at such an amazing center for translational research, we've started a large program called the Clinical Genomics Program, which enables us to take children with all sorts of genetic deficiencies and abnormalities, similar to the boy in the bubble is the famous one everyone might relate to, and probe into what the gene defects are, and then look at the pathway that's involved and try and come up with an intervention. And that's been very successful, very exciting, and led to a number of clinical trials and actually patents and involvement with companies in new drugs because of the desire to not just find out what's wrong, but actually fix it.
And so you've used some terms which I'm very familiar with, which a lot of people may not be. It's sort of the bench to bedside, it's the lab to market, translational research. So when you think in those terms versus the majority of the scientists within NIH who might be doing pure basic research, what's the difference between the translational versus the pure basic research?
Well, the translational involves the second half of the virtuous circle of discovery. So the first half is getting samples or data from patients, going into the lab and doing molecular experiments or biochemical experiments or cellular experiments and figuring out, okay, this is what we think the disease process is doing. But there's another half to that, and that is now going back from the laboratory to the bedside to the patient and saying, look, this is what we think is wrong, and here's what we're going to do about it. And so that involves a whole nother set of requirements. So there's, first of all, you need a clinical team. You need nurses, you need physicians, you need people that are case managers, people that do regulatory. And there are regulatory requirements that you have to comply with that involve patient-based research. So it is much more sophisticated at some level than the pure discovery work, but it requires a certain environment where you will get the support to do that. And I think the NIH intramural program, certainly the Bethesda campus, is really an ideal place to do that. And so for me, someone who you might say early in my career abandoned clinical medicine because I was so fascinated by science and wanted to do fundamental research, allowing myself to get back into the game using my research to push new therapies has been facilitated by this amazing environment.
And then basically I looked a little bit at your bio, but there's numerous publications, a lot of personal research and patents. So talk a little bit about those things that you have done that were, I don't want to say worthy of patenting, but got enough familiarity that they thought there was something there that might be able to be patented, that might be able to be translated or commercialized in the future. Talk about those things that you're most proud of that maybe have been patented that have the greatest potential for translation.
Well, I think one of the most exciting things we discovered early on was the fact that we could trigger T cells that were pathogenic and causing autoimmune diseases to die using the specific antigen that triggered those T cells in the first place. And that was a very new concept that a lot of immunologists had trouble understanding and dealing with. But we were able to patent it and actually get a lot of support, which is still to this day work that we're doing to do clinical trials to show that that could ameliorate autoimmune diseases. So that was a very exciting thing. Since that time, we've had a number of different therapeutic approaches for different kinds of diseases, usually genetic diseases that we've discovered that the NIH has patented. And I think it's very interesting. It's actually part of your responsibility as a federal employee to comply with the Bayh-Dole Act, in Executive Order 10,096, to bring to the patent officers of the NIH and the patent technology transfer team things that you think are innovative and should be commercialized, and then they make a decision. Not everything that we thought might be commercializable they agreed with, but that's part of our job to do that. And the payback is that it does then force you to think more about how you're going to apply what you've learned from your basic research. And also, if it is commercialized to the point of being licensed, that money flows back into the NIH coffers to support more research. So the commercialization process actually pays dividends to the taxpayer to support additional research that now is not coming out of tax dollars, but coming out of money that's been earned by those discoveries.
I compliment you. You've explained that very well.
Oh, thank you. Thanks. I'm sure you know more about it than I do.
No. But I mean, to be honest with you, a lot of the scientists, and it's really, there's multiple missions within NIH to advance the quality of healthcare, to advance science. But Congress also likes the one that says, try to find a way to get a return on the $40 billion that gets invested in NIH
That's
research,
right.
Correct?
That's exactly right.
And have you gotten close to any of your research or patents getting into the marketplace yet?
Well, we have right now 2 clinical trials we're very excited about that are medications for genetic disease we've discovered that look like they're working very well. And I think those will be FDA approved, and therefore then they would be sold as medications. So yes, we've taken the circle all the way.
And I think what you're talking about is internal clinical trials within NIH rather than externally. And there's listeners probably that understand that. NIH has the ability to fund internal clinical trials without having to work with a pharma or bio company or an external firm to do clinical trials externally.
It's a little more complicated. So generally, clinical trials for rare diseases have multiple sites. So in both cases, there are sites. One of the sites is the NIH Clinical Center, but there are other sites in Europe and other countries involved externally.
Gotcha.
And secondly, these are done in close collaboration with pharma. So pharma is providing some support, NIH is providing some support. So it's really a public-private partnership. And I think there's great strength in that because it enables you to meet the practical reality of the marketplace. It enables you to meet the practical reality of what it takes to get an FDA approval. And pharma companies are far more experienced and knowledgeable about those sorts of things than an NIH investigator. And so to me, building bridges, levering resources is really the way to go. And so that's how these are.
And it also provides the validation for the research you're doing.
Yeah,
and it also, as you mentioned, leverages the public dollars with private dollars coming in, which enables it to go further.
Absolutely. Absolutely, Rich. That's exactly right.
And with all of the success you've had with your research, I understand yesterday you were just named or elected into the National Academy of Medicine.
Yes, that's true.
Congratulations on that honor.
Thanks very much.
And for listeners, tell us a little bit about the National Academy of Medicine and what it takes to get into it.
Well, I would describe it as a recognition by your peers, and peers in the sense that it's the members, the current members, who elect new members. So it is the people who themselves have been judged as being outstanding biomedical researchers selecting, in this case, me, to join their ranks. So it's a great honor, and I'm glad that these very illustrious peers of mine around the country feel that my research is worthy of that honor. I think it's great. I don't know much about the organization yet. I've only been involved for one day. So, but I'm sure I'll learn more.
Well, it's nice to have a newly minted member.
Thank you.
Doing his first external podcast on BioTalk right here in your backyard.
I hope it won't be the last one.
I hope it's not the last one, because once we get to phase 3 clinical trials and that gets into helping patients, then we'll bring you back again.
Sounds great. Sounds great.
So let's talk about why we're here today. Your name was brought to me by Randy Bellata, who is the president of the International Biomedical Research Alliance. And she said, you're a fantastic scientist, researcher, and also involved intimately in that international alliance, and it involves a scholars program. So let's talk a little bit about how you met Rhonda, how she met you, what you think about this International Biomedical Research Alliance, and let's talk to the listeners about this.
So we at NIH historically have been largely an educational institution for postgraduate researchers, so at the bench researchers. And it was really Harold Varmus towards the end of his directorship of NIH that got the idea of opening up the government labs to students and getting top students to come there to work and understand what the NIH is all about, what the federal mission is, and also go on and have great careers as investigators. And he called me and a few other people over to Building 1 to talk about how we could do that at NIH, because NIH, as you know, is not a degree-granting institution. And I had known Harold because we connected at MIT before I got to NIH. And so we talked it over, and one of the ideas that came up was to create public-private partnerships between the NIH research laboratories and educational institutions that had graduate programs. And although the focus, I think, of our discussion was mainly on American institutions, I had the idea, let me just say that one of the things he emphasized was we don't want to try and reduplicate what's already amazing universities around the country are already doing to educate graduate students. We wanted programs that were doing something different beyond what was on offer at the moment. And so one of the things I decided to do in the wake of that conversation was to take a look at what's going on in graduate programs across the country. They seem to be great. What are the not-so-great aspects? And it turned out there were more not-so-great aspects in American education than I thought for graduate students in biomedical research. So first of all, one of the big problems is Today, science is international, collaborative. It's usually not what we think of from the movies of the 1950s, where there's one person in a long white coat sitting in front of a microscope figuring out what's going on with his cells. It is dynamic interactions between scientists in other countries, scientists in different disciplines, and we pay a lot of lip service to that. But the structure of our graduate programs is putting a student in one laboratory in one institution where they labor away, and that's what they do. So it was missing critical aspects of how to train students to do contemporary science. The other thing is that the idea of interdisciplinary work wasn't really there given that model. And that model, this is the master-apprentice model that goes back to the Middle Ages. In this country, the model hadn't changed at all since it was set up sort of around World War II. Other aspects of the model that were a problem was in most graduate programs, you come in and do 2 years of rotation and coursework. It's all boilerplate. Every student does the same thing. So it doesn't matter if you studied biophysics in all your classes in college, you might have to come in and take a biophysics course because that's what the curriculum is. Most of the top students have already been in laboratories. Why they need to do 3 more rotations to sit around labs when they're not really working on their thesis. So we thought they were inefficient. And finally, one of the biggest problems is that it takes forever to get a PhD in this country. At the time we started the Oxford-Cambridge program that I'm going to talk about in a second, the National Research Council had calculated that the average time to degree for a biomedical PhD, I'm not talking about English literature or philosophy, was 7.8 years. Now we knew, the NIH knew a big problem was independent investigators were not getting their first foundational grant for their lab, which is called an R01, is the lingo, until they were 42 years old. But no one ever went back and said the reason they're 42 is that they spend 8 years getting their doctorate, then they have to do their postdoctoral work, and then maybe they can finally look around and get their own faculty appointment. So it was really bogging down the system by keeping students in labs. And why were they in labs? Well, the single person who's most conflicted at letting them out of the lab is their principal investigator, who's their mentor, who gets to make that decision. And
who wants to keep them there.
Wants to keep them there. So all of these things together suggested that we needed a new model. So what we decided to do is, first of all, let's make it international by definition. So let's pick degree-granting institutions outside of the United States. And when you look around and think about the practicality of students doing theses in other countries, really, England makes a lot of sense. And really, if you look at the international university rankings, Oxford and Cambridge, which are our partners in this enterprise, always are in the top 5, competing with Harvard and Stanford and other institutions. So those seem like a natural place to start. Maybe that's not the only ones we would ever have, but that's where we started. And so then how do you do that? How do you have mentors over there and mentors at NIH? So the idea was that a mentor or mentors from the UK institutions and a mentor or mentors from NIH had to form a collaborative project. And the student would be the glue who would do part of their time, roughly 50% at NIH, and roughly 50% in a laboratory at either Oxford or Cambridge. Now, some of the students are very enterprising, put together very complicated multidisciplinary projects. They might have 4 mentors that they work with, but they had to have at least 2, because that's the definition of a collaboration. You got to have at least 2 to tango. And then we learned a lot about how this works. And the other thing I should mention, just as an aside, is that we just totally got rid of this mandatory boilerplate coursework and rotations. So students come in, they spend 2 months in the summer writing a project outline, reading the literature that's relevant to their research. They start on their thesis on day 1. If they want to take courses, we have courses at NIH, we have courses at other institutions in Maryland. Oxford and Cambridge has a whole suite of courses. So they can take courses, but they do that pari passu with doing their thesis work. So they're already on the march to finishing the day they start. And by an agreement between the universities and the NIH, we decided the target time frame was going to be 4 years, so roughly half the time. And I'm happy to tell you, for the first 230 graduates over the last 20 years, it's coming in about 4.1 years. So not everyone finishes in 4 years, but some finish in 3. Some maybe have a project takes a little longer, go to 5. But on average, we're hitting the target right on the nose. And we learned a lot of very interesting things about education from this project. So one is, and this is probably the most heartening, is that very top students want to do something like this. They find it more exciting than the traditional models. They find it more challenging. and they learn interesting things. So what do they learn? Well, first of all, if you imagine the triangle of two mentors up on top and the student below, these two mentors might be leaders in their field, huge egos, very convinced that they know the right answer. But the point is that there's two of them. So now someone's got to put together these kinds of inputs and then actually take an action, do the next experiment. So the student becomes very good at, first of all, politically dealing with these two individuals, but on a scientific level, filtering what the inputs are getting and making a decision. When you're a practicing scientist, you do this all the time. I go all around the world and present my science. I get questions. I get criticisms. And there's sometimes from very prominent scientists whose opinion I respect a lot. I have to now filter that in with my own knowledge, make a decision, and decide what we're going to do in lab. So the students are getting this from day one of graduate school, how to act like a real scientist and take diverse points of view and put them together and then move forward. The other thing that we noticed is that the atmosphere, the environment of science in England and in America is very, very different. I'll just give you a very cute and quaint example. In most prominent institutions in Britain, certainly at Oxford and Cambridge, at 3:45 in the afternoon, so just an hour or so before we started this podcast, everyone in the lab puts down their pipetting device and goes to the tea room and sits around for 45 minutes and has tea and cookies or coffee, whatever, and they talk. It's not a group meeting. There's no data, slideshows. There's no agenda. They just talk. Now, sure, it could be about Brexit. It could be about the latest movie. But in my experience, having spent some time at these universities, a lot of times it's about science and ideas. And everyone else is in the room. You can bump into other scientists and talk to them about, hey, how'd that project go with the seminar you gave last week? So it introduces a level of interaction and thought and discussion that I can tell you is almost impossible to get in a United States lab. If you try in your lab in the U.S. to get people to stop the frenetic rush for data and actually sit and talk, it's very, very difficult. Now, because of this experience with this program, I've implemented this on Friday afternoons. I can't do it every day. It would be impossible. So every Friday at 3 o'clock in my lab, people have to leave the lab and go down to a local little conference room and sit around and eat cookies and drink coffee and talk. And I can tell you at 3:59, they're out of there. And it's just a cultural difference. And I think having exposure to both points of view, the drive for data, the drive for success, the drive for accomplishment, combined with a more contemplative, discussive approach to science is very, very healthy. And I think the students, having experienced this, come back and tell me that they feel it is true. And some of them have tea time in their own labs now. So I think it's beneficial. So it's a very different kind of a program for very accomplished students that want to move quickly into their research careers.
So we need to do more tea times. I'm a tea drinker. So
well, there you go, Rich.
I would fit right in. Invite me in someday for 3 o'clock. But I guess at the end of the day, your passion is coming through for this because it's something I guess you've been involved with for the 20 years.
Yeah. So I started the program.
You started the program.
The first class was in 2001. And we are very excited that it has really been successful in attracting top students, many of whom now are in their own research careers. A surprising number are actually in biotech or pharma. I've been astonished that the number of students that have started their own biotech companies is the same as the number who've become academic assistant professors. That shocked me. I thought most of the students would become academicians. Are most of
those doing that in the UK or over here?
It's interesting. Most in the U.S., but I would say between 10 and 15%. So these are mostly American students that are participating in the program, do wind up staying in England, which I find fascinating.
Really? That's
also
interesting.
Yeah.
And then what is the relationship with the International Biomedical Research Alliance then?
So as you alluded to earlier, I don't know if it was on the podcast or before, the NIH is a government institution. It has very strict kinds of regulations, what you can spend taxpayers' dollars on, how things are structured. And it became obvious to us very early on that this could not run strictly as a governmental program. And many of the programs that have been started for training at NIH end up just basically being a hiring mechanism for the lab. And we didn't want to take the United States' most elite students and just put them in labs. We wanted them to have a rich program of experiences that complements the scientific training. For example, exposing them to careers in industry, exposing them to thought leaders in different parts of research, whether it be academic or whether it be pharma or whether it be even government. And to do that, we really needed help because we need people who know that space, who can advise us, who can participate, who can support it. And so over time, a group of private individuals have started the International Biomedical Research Alliance. They've been very capably guided by Randy to create a 501(c) organization in the district. And they've just done a marvelous job being our partners along with the universities and helping these students to have an amazing experience. And the students have been extremely grateful for everything that they've done.
So they're an intermediary to sort of manage this private-public partnership between the public and the private worlds.
Yeah, and also bring in a new perspective that's really beyond the academic perspective. Because the universities, of course, are very focused on academia. The NIH is kind of focused on research aspect. And so these kind of open up the students' eyes to the whole way in which biomedical research interfaces with all of society. We have lawyers on their board that have to come and talk to the students, people from finance. So I think that has allowed the students to have such a unique experience that's beyond what most graduate students would experience in U.S. or U.K.
Yeah, well, it sounds very interesting and probably a way that we can talk offline where biohealth innovation can get involved since we are a private-public partnership ourselves, working with academia, industry,
Absolutely.
Financial institutions. And I'm sure there's something we can do right in our backyard for collaboration. It's just getting to know one another.
Exactly.
Which is what this podcast is doing for us.
Thank you. Thank you.
You already talked about the success of the program, which is pretty impressive, 230 graduates and their average 4.1 years versus a 7.8. Let's talk about some other things that you see in the future for NIH, NIAID, and maybe the Scholars Program you have. What are some of the challenges and opportunities, both positive, negative, that you see in your field that you're going to encounter in the future based on what's ahead of us? And research, economy, I don't want to talk about politics. We're not going to go there today, but it does have an influence. But, you know, one of the things that's interesting is the NIH budget has continued to grow over the last couple of years.
Yeah, and I think one of the great things about what Francis Collins has done as a leader is that rather than have a huge doubling of the budget, something that is very impressive and gets a lot of headlines, he's taken a more gradual approach of asking for the kinds of increases that will meet inflation, that will give us a little bit of opportunity to explore new areas or take advantage of new potentiality in research that's uncovering opportunities, and use the money wisely and very deliberately. I think when the NIH budget doubled in the early 2000s, in a sense it was great, but there was sort of a boom and bust phenomenon where once Congress did that, they felt like, okay, we took care of you guys, go away and don't bother us. And that's a very natural reaction. But the problem is needs grow every year. And if you totally expand the system, then you've got now an expanded system that still is going to have bills to pay with inflation and so forth. So I think the approach has been a wise one. It's been a deliberate one. It's been focused on diseases that people care about. And I think that's been really terrific. When people raise the question of future prognostication, I always smile looking back on what I've seen. And I came from a background in molecular biology where we did DNA sequencing on 80-centimeter gels by hand, and you would read the bases one by one from an autoradiograph. And the idea that sequencing that might have taken months to do now happens in a fraction of a second is something no one could have predicted. So I think that the advance of technology, the advance of insight, the opportunities for conquering new diseases just are going to grow in ways that, honestly, Rich, you and I sitting here today could not possibly know 20 or 30 years from now what it's going to look like. I can tell you some things I see right in front of us. They're pretty obvious ones. I certainly think that immunology, its impact on many different organ systems in the body is becoming clear. And even now people are talking about neurological diseases, neurodegenerative diseases having an immune component. We've certainly learned in the past few years that cancer can be tackled to some degree by changes in the immune system. I also think that some of the fundamental advances in genome editing are so incredibly exciting. The idea to, in my area of immunology, correct genetic defects, at least in somatic cells. Germ cell changes, like what happened in China, I think we have a lot of thinking to do before we go that route. But I think certainly altering somatic cells to improve their function, for example, immune cells that can correct a genetic deficiency, those are things that are not going to affect the germline. I think they could be relatively safe. We have tools now that should be explored, I think that will be explored to use that as a new approach. And again, that's something that even 10 years ago would have seemed like science fiction.
And so as we're talking to STEM students who are looking for things to do in the future, even with the advance of technology, there's still going to be a big role for a bench scientist.
Absolutely. And I think the funny thing that you run into with students, they're so in awe of what has been done. DNA structure's been solved. DNA sequencing has been figured out. Immunotherapy has been figured out. And they get into the mode of thinking, gosh, is there anything left? Have we done it all? And I think you have to remember back to so many times in history when a scientist has said, we've figured out how the universe works. And then along comes Einstein and quantum mechanics and the relativity theory, and everything gets upended. So we have to look at science and look at ourselves as human beings and realize that our ability to see the future and understand all the world around us, whether it's the biological world or the physical world, is so minute compared to what's out there that there are just going to be endless things that human beings will be able to discover with enough ingenuity and hard work. And so I think there's plenty for all the STEM students to do
in the future
going forward. And hopefully NIH will be able to fund a good part of that.
We're talking with Dr. Michael Lenardo, who's Chief Molecular Development of Immune System Section for NIAID, National Institute for Allergy and Infectious Disease at NIH. And we've talked a lot about the scientific background, but I understand there's another side to you, which a lot of people don't know, and a few people do know. And let's talk about your musical interest.
Well, so I think you're referring to the NIH band, which includes a really exciting group of talented people. I would say I'm probably the least talented person in the band, but it goes all the way from students who have only been in NIH for 6 months, coming out of college as a postbaccalaureate student, up to the director of the NIH, Francis Collins, who himself is a very, very talented musician, that come together and do songs that are interest all of us. And we perform basically as a cover band. We do a lot of NIH events, some charity events for the NIH. And it's been a lot of fun. It's a great group of people. I can think of, for example, Dr. John O'Shea, who's sort of my vintage. We like a lot of Beatles songs and Bob Dylan and things like that. Some of the young students have us doing Adele and Outkast and other songs. So it keeps you fresh. It keeps you on your toes. You got to learn a lot to be in the band. And it's really a lot of fun. And I think judging from the response of the people that come to hear us, I think people enjoy it and have a good time and have fun dancing. And
so you like Times Are A-Changin' and you like the movie
Oh,
Yesterday.
Yeah, of course.
Did you see the movie Yesterday?
I have not yet seen it.
Oh, you
I've
have
been
to
busy
see
in the
it.
lab, but I will try to get to see it.
It's very interesting about the Beatles.
Yeah. I'm the vocalist for a couple of the Beatle tunes. So,
Oh, are you?
Yes.
What are your favorite songs?
Well, the one I do in our most recent gigs is Hard Day's Night. Francis Collins and I do 8 days a week together,
really?
Harmonizing. So
I'm not going to ask for a little bit of that right now because there's no musical accompaniment for you. This is really interesting. We've digressed a little bit, but it's a very interesting digression. Let's talk, as we close here a little bit, Dr. Lenardo. Talk about some of your personal goals. You've accomplished a lot, especially with this latest recognition with the National Academy of Medicine. What are some of the future personal goals that you have that you'd like to achieve?
Well, I think I'm very devoted to the lab and to the kind of research we do. I think being at the NIH in the intramural program, we have sort of two side-by-side missions and responsibilities. One is to do the highest level research we can, particularly as it pertains to patients. And so we have a lot of goals in the lab related to that I personally want to accomplish. And then I think I've also spent a fair amount of time talking about our training mission and the idea to create the next generation of scientists who are going to carry on the work that we do. The intramural program has a very illustrious history. Many Nobel laureates have worked there, have discovered important things within the intramural program, within the walls of the building that I occupy. And I think we want to create the next generation who will continue to carry on that research. So I think those are my goals, to fulfill those two missions of the NIH intramural program.
Very admirable. And so as we close, what are your words of wisdom for the emerging scientist that would like to evolve to where you are today?
Well, I think going back to a theme that I mentioned a little earlier, I think if there's anything I've learned about science, and that is that there's a lot more that you don't know than you know. And I think many of us, particularly if we were good students in college, good students in high school, we sort of get rewarded for having an answer for everything and always knowing the right answer. But when you get into research, particularly as it relates to patients and things that can affect people's health, you realize there's a lot you don't know. And you have to be very humble and learn from your mistakes, learn from your failed hypotheses, and get used to that, getting used to the fact that you're going to get it wrong more times than right. But if you hang in there and work hard, you can get the right answer. And if you do, it can have a big impact on people's lives. So I think humility is probably the word that I think of that's most useful for a scientist to know.
And one that wouldn't be the first one that people would think about.
They may not. I mean, there's other ones too. I think being adventuresome. I mean, you're really looking for
curiosity,
regions unknown in the intellect and in the body and in the world of science. But I think humility is a good place to start.
Well, I think it's a good place to start and a good place to end in our podcast right now. And I'd like to follow up with you because this has been very interesting and stimulating, and I'm sure the listeners are going to enjoy it a great deal. Maybe we can follow up with some students and do.
I think that would be. I think you'd be
have the interaction
really
with students and
have
you
a lot of fun,
on a
Rich,
podcast, which would be fun to do. But I'm going to close and thank Dr. Michael Lenardo, Chief Molecular Development of Immune System Section at NIAID at NIH, for coming on to BioTalk today.
Well, thanks for inviting me, Rich.
We're definitely going to do another one in the future.
Great. Thank you very much.
Thanks for listening to BioTalk with Rich Bendis.